Christine's going to tell us about one aspect of this work, where they've tried to engineer a specific part of photosynthesis and got some very interesting results. Okay, so without any further ado, I'll hand over to Christine.
Thanks very much for the introduction, John, and the opportunity to share our paper that was published in JXB earlier, I think this year. So without any further ado, as they say, I'll just press on. So the goal of the work that we were doing with the ictB protein was to improve photosynthesis through its introduction into tobacco. And what you can see on the image here is just a campus here at Essex, which I always take great pleasure in sharing with people because it's very nice and it looks a bit like that today. So moving on. So I guess the question we can go back to is, well, what's the need to increase crop photosynthesis and crop yields? And if we look at improvement in crop yields in the 20th century, this was brought about effectively by the Green Revolution. Traditional breeding of desirable traits, such increases in harvest index, and in addition to which the application of chemicals, which really improved yields dramatically. But what's happening now is that the increase in yields of major grain crops is now slowing down. The yields are not declining, and I think this is a really important difference. The increase in increases, the year-on-year increases that we need to feed the growing population is now slowing. As you know, the population's increasing, predicted to be 9 billion by 2050. And something I won't really talk to you about today, but obviously our climate system's changing through increasing CO2. So if we look at the growing population, this is obviously a prediction. We don't know for sure which way it's going to go, and this little graph kind of gives you what it may look like. And in actual fact, this is kind of predicting where it might be, I guess at the bottom end of the predictions, may even be as high as 11 or up to 13. I think the bottom line is, whatever one of these numbers you pick, we're still going to need a significant amount more food in order to feed the growing population. And you can see the predictions at 2050 sit around about 9, but by the end of the century, this could be substantially more and is likely to be. Okay. And then if we look at yield increases, as I've already mentioned, these are not on target to meet what we need by 2050, let alone what might happen later on in the century. And if we look at the major grain crops that feed the world, and just pick on wheat, because that's a major crop in the UK, and we follow this bright pink line, we can see the yield in tonnes per hectare as we go forward. If you look at the solid line, as we go along here, this is, if we keep up the yield increases at the same rate, this is where we will be. But the dashed line shows you where we really need to be. And that pattern is the same for all 4 of these major crops. So you can see that we've got a major gap. So I think given the combination of the requirements to find strategies to increase our yield year on year, and the growing population, then I think it really is a pressing issue. So we need to look at all approaches that we can take to deliver this. So why target photosynthesis? And I'm not going to talk in too much detail about this, but I think it's worthwhile just to highlight a few points. Photosynthesis ultimately determines crop productivity. But surprisingly, photosynthesis rates have not been used to identify high-yielding lines in the past, and there's a very good reason for that. It's actually relatively difficult. What do you measure? When do you measure? How frequently? And I'll highlight that perhaps as we go through the paper later on in the presentation. But one key thing is that the ability to convert the light trapped in photosynthesis into a product is not attained, and that maximum energy conversion efficiency is not attained in the field, and therefore there's a gap in this as well. And therefore, by manipulation of photosynthesis, with the opportunity to close that. So when I'm talking about improving photosynthesis, I'm talking about the C3 cycle, the Calvin-Benson pathway. And plants using solely this pathway are the dominant plants on Earth, some 85% in terms of numbers of species. And I think an important point is that this includes the major crops such as wheat, rice, potato, and soybean. And there are now a number of examples of successes to improve photosynthesis in C3 model, it should be model species, not model crops, in the greenhouse, and some field successes that have been published recently. So I think not only the theory would indicate that we can improve photosynthesis and therefore yield, but we also now have very clear empirical examples of this. And so improving the Calvin-Benson cycle, this is a little cartoon diagram of the Calvin-Benson cycle. We can see up at the top, rubisco that fixes CO2 in from the atmosphere into 3 carbon compounds, which are then shuttled through in the regenerative part of the Calvin cycle into this CO2 acceptor molecule, ribulose 1,5 bisphosphate, and the cycle continues. Just on passing, an important job of the Calvin cycle is to fix CO2 from the atmosphere into compounds, which go off to produce a whole array of molecules that then feed in to the pathways of isoprenoid biosynthesis, starch, sucrose, shikimate, thiamine, and nucleotides. Don't have time to talk about that today, but of course the other job of the Calvin cycle is to sustain itself. Now, over the last few years in my lab, we've targeted a number of points in the cycle, and you can see these highlighted here. And I'm not really going to talk to you about that today, but ictB was a protein that was identified in cyanobacteria and thought to be able to shuttle additional CO2 towards rubisco and therefore improve photosynthesis. And I'll just talk you through a little bit about the ictB protein, which is the subject of today's talk. So, features of the ictB protein: it's native to cyanobacteria, it's not a plant or algal protein. It's 52 kilodaltons, and it's a membrane protein with 12 spanning domains. It has a no antigen ligase domain. It's highly conserved across cyanobacterial species. And it was originally targeted or identified from analysis of a high CO2 requiring Synechococcus mutant. And this gave the hypothesis that ictB was involved in inorganic carbon accumulation. It's now known that the ictB is not a bicarbonate transporter in cyanobacteria. So really, the function of this protein remains somewhat of a mystery. So the very first work that was done is, and please forgive me for this diagram, I could not get a good image of this paper for one reason or another, but this was the work that was done some time ago now in 2003, where the ictB protein was expressed in both Arabidopsis and tobacco. And what was observed was, I think this is the tobacco data, there's 4 transgenic lines you can see here, and wild type is the filled in triangle. And if we look at the response of photosynthesis to increasing CO2, we can see this is the wild type line here. These are the rates that you observe in the wild type plants, and those plants overexpressing this ictB protein, there was an increase in photosynthesis, both in the section of this ACI curve that shows limitation by rubisco, which is in this region, but also limitation in response to electron transport and IUBB regeneration. So it was quite clear that this was, from this data, that there was a very positive effect on photosynthesis by introducing this protein. And then a few years later, in my lab, funded through BBSRC, we published a paper in JXB where we'd overexpressed ictB, and our interest in this was to see if we could get additive or synergistic effects by overexpressing other targets in the Calvin cycle. And just as a way of background, one of the things that we saw, which had already been shown by the previous group, was that ictB alone was able to increase substantially photosynthesis. But what we didn't see was any increase in this part of the curve that would have been due to increases in activity of rubisco. It was mainly in the region of the curve where electron transport and IUPB regeneration were involved. And then we looked at the total biomass, and we can see with the ictB lines, we did get an increase in total biomass relative to wild type. What we were interested in was the synergistic effect with combinations of these different proteins. But for today's talk, so this really supported the work that had been done some years earlier, but we extended it by taking a multi-gene approach. And then Will Hay and Steve Long's lab added ictB into soybean. And when he looked at the photosynthesis rates, we can see here, this is the wild type in black. These are modeled based on the data. You can see in the red curve that there was increases in photosynthesis in the ictB lines, and this is shown below as well. So if we move on, this is in the field, the data from the field also supported the increases in photosynthesis and also some increases in biomass. So moving on. There's two other papers, and what I'm doing is building up a picture of the positive effects that had been observed previously with overexpression of ictB. This was around about 2015. Gong et al. Published a paper where they'd overexpressed ictB together with a bifunctional cyanobacterial enzyme for FBPase, SBPase, and they saw increases in photosynthesis and mesophyll conductance. And then, but they also showed that it was not localized to the chloroplast. So if it was helping with movement of bicarbonate, it certainly wasn't doing that in any close vicinity with rubisco. And then more recently, work in a C4 species where ictB was shown to increase grain yield in ZMAs. I think the surprising, this data was really nice, and in the maize work, because what they did was grow the transgenic plants in a number of different sites and showed that from year to year and under different conditions, the response to the overexpression of ictB was not always the same. So in other words, sometimes this provided an increase in biomass, and other times this was not observed. So some level of environmental sensitivity to this overexpression. And so coming to the actual paper that was that we published earlier in the year, this was funded under the Wright program, realizing increased photosynthetic efficiency. There's a number of partners here. It's headed by Illinois, and you can see all of the partners down here contributing to this program. And we all have our own focus, and modeling is the overarching part of this project, where we have optimizing canopies, algal mechanisms, improving rubisco, and I'll let you read the rest yourself. What I've been working on is RUBB regeneration, and that's our focus currently at Essex. And what we're doing is using these manipulations in order to advance translation to provide improved crops for Africa. So we can come to the paper now. So this was, as I say, was published earlier in the year. And what we did was to take our ictB tobacco transformants out into the field in Illinois. So this work was started in my lab by Andy Simpkin, who's on the paper, and also contributions from Hussein and from Chidi, co-authors working on the molecular protein aspects, and Kenny Brown, who actually was due to go and do the field study, but because of the COVID situation, we were unable to do this. But the two lead authors on this paper, Ursula and Liana, stepped into the breach and did an absolutely excellent job, not only in gathering the data, but also in putting together this paper. So great thanks to them for a really excellent piece of work. So let's just go now through the data that was published. It's been notoriously difficult to identify and find the ictB transcripts in transgenic plants, and most authors have recognized this. And in actual fact, if you look at the Mays paper, they've gone to great lengths to be able to show this. But what we did was just take a look at the transcripts up at the top. This is just a semi-quantitative RT-PCR, and we've got a quantitative RT-PCR qPCR below. These data confirm the work of Andy Simpkin, and because these were exactly the same lines used in the field as he studied in his paper. And when we go over to the field trial, here is the design. We had 4 ictB genotypes, 1 azygous, which was mixed, that had come from each of the independent transformations, and a mix of a number of independent wild type plants. There's 12 replicates, as you can see, and 4 plants per rep. In total, we had 288 plants with 48 per genotype. And measurements were made. There was really a thorough analysis of aspects of photosynthesis, including ACI curves, AQ, and diurnal measurements. Biomass was looked at, leaf number, leaf weight, stem weight, number of leaves, and we'll go through the paper to look at these data. So if we look at the first set of data that's presented, this is in figure 1 of the paper. What we've got here are what are called AQ curves. This is a response of assimilation rate to changes in light intensity. And measurements were made in August and again in September. If you look at the August measurements, wild types in red, the transgenics are either on the same line as the wild type or below. In September, you could persuade yourself there might have been some small increase up at the top of the curve in 2 of the transgenic lines. But when we take the season average, there was no significant difference observed. And so this was somewhat, I guess, as we went through the season, somewhat disappointing to us. And just to be sure, we then followed this up by ACI curves. So this is the assimilation rate against increasing internal CO2 concentration. And you can see once again, very, very similar pattern to that what we saw with the AQ curves. Some of the transgenics just below the line in August, and possibly 1 or 2 above the line in September, but the season average coming up with no significant differences. And this may be highlighting that there is definitely an environmental and maybe even developmental impact on the improvements we see in photosynthesis in the ictB lines. And then we go on using the ACI curves and looking at VC max, which gives us the maximum capacity of Rubisco, and looking at the apparent Jmax, which is electron transport and IUPB regeneration. The red is showing you the wild type plants, and the 4 different shades of gray are showing you the different transgenic lines. And you can see there are some small differences between the different transgenic lines, but really there's no significant difference, certainly no significant increases in any of these parameters relative to wild type plants. Now, down at the bottom here, we have a measurement of what would be the compensation point. And what we can see again is there are no significant differences. And this might have been a parameter that you would have expected to see the differences in, particularly if this was a bicarbonate transporter of any description, enabling ribisco to function at lower levels of CO2. It's just to be sure that we didn't miss any significant changes in CO2 assimilation. What you can see here are diurnal measurements of photosynthesis. This is taken over the day. Often in the way that we can see differences during the season, you often can see differences in a daily pattern of assimilation, but really this is the assimilation rate we can see here, and the wild type again in red, and you can see there is really no major differences, or certainly no positive differences, between the ictB lines and that of the wild type, and none of the other parameters measured, such as stomatoconductance, internal CI concentrations, or the intrinsic water use efficiency was not really majorly changed in these lines. And then looking at leaf carbon content and leaf carbon isotope composition, there was no significant differences seen between the transgenic lines and that of the wild type. So I think what we see here is really a thorough analysis of major parameters of photosynthesis, that we can see really no major differences that were consistent or sustained over the season or over the daily cycles. And then obviously the proof of the pudding is in the eating. Can we see more above ground biomass? Not likely, really, you would say, given that we didn't manage to pick up any significant changes in photosynthesis. But nevertheless, this was looked at across all of the genotypes. And the only parameter that was increased, and if we look down to the bottom left-hand side here, the total number of leaves were increased in all of the transgenic lines relative to wild type. However, in no other parameter, if we take the total above ground biomass, if anything, in 2 of the lines, there was a decrease. And stem dry weight, no increases, leaf dry weight, and so on. So total leaf area wasn't significantly changed. So although we had more leaves, it looks like they were more than likely smaller than the ones in wild type. And with some transgenic manipulations of metabolism or photosynthesis, we have seen changes like this, where you get a modification of the development of the leaf, but there is no increase in biomass. I think we got quite excited at one point when we thought perhaps the number of leaves had increased, but in fact did not feed through to increases in biomass. So just to summarize this, ictB has been shown to increase photosynthesis and yield in greenhouse grown Arabidopsis, tobacco, rice, and soybean. ictB has also been shown to increase photosynthesis and yield in field grown soybean and maize. But in our case, no increase in photosynthesis or yield was seen in the field grown tobacco in Illinois that particular season. So where are we with this? What's the future prospects for ictB, and what questions remain? I think we cannot possibly rule out that ictB could be beneficial, for example, in controlled environment conditions, and or in combination with other manipulations. And given that the tobacco field study was only one season in one location, and the data that's been obtained from maize and soybean, this would indicate that perhaps environmental conditions could be very important, and therefore it wouldn't, we shouldn't give up necessarily on ictB. I think the big problem with it is, of course, is that the function of the ictB protein remains unknown. And Hussein Gerli tried to begin some work on that and try to express the protein so that we could start to tease apart perhaps what this protein was actually doing. But if you look at the maize paper that was published at the end of last year, you will see that they have tried to tackle this question also, and ended up having to express the ictB protein as peptides. And they still have not really nailed what this protein is doing. I think they identified that it might bind. They've done some in vitro assays where they asked the question, can it bind to proteins in the cell? And have identified some chloroplast proteins that it may interact with. But that's a puzzle to me, because I'm not entirely sure how, if something is expressed outside of the chloroplast, and in fact it's in membranes outside the chloroplast, how it could be interacting in that way. But I think it's an interesting point and is worth further questioning. So it really is an issue. How do we detect this ictB protein, and how do we take it forward? But I want to leave on a positive note. I think there are opportunities for using this protein, as I've said just a few seconds ago, under controlled environment conditions, if we think of vertical farms or in horticultural situations, but also in combination with other proteins. But a really key thing when we do transgenic work is often we do not have the opportunity to do multi-site, multi-location field trials. And I think this is really something that will be needed in the future in order that we can really have the opportunity to test out potentially interesting and positive manipulations we see in the greenhouse. So I think with that, I'll just thank everyone for coming along and listening to my talk today. And I'll stop sharing my screen, I think, so that I can allow John to share and ask or shuttle the questions to me anyway.
Right. Thank you very much, Christine, for this very clear explanation of the paper and the background. What I liked about the paper was that it was a really neat case study of the problems that we are likely to face in translating lab-based or greenhouse-based results into the field. And also, it really demonstrates how important the environment is going to be in determining the phenotype of these sort of plants. And I think this is a message that many of us who are working in this area have come to realize. And this is a message that younger researchers need to take on board. So yeah, thanks again for this very clear presentation. Okay, so now we can open up for questions. If you're online, you can either raise your hand. It would be nice if you switch on your camera so we can actually see you asking a question. But you can also ask a question through the chat function. And initially, we'd like to encourage any early career researchers in the audience to ask their questions first.
So there is one in the chat, John.
Yeah, okay. Okay, so we have a question from Haim. Hello, Haim. So what do you attribute the lack of difference in growth and photosynthesis compared with past work to the field conditions, the thoroughness of the physiology study, or is it perhaps tobacco being less limited in the field compared with other plants? So that's Haim's first question. Do you want to tackle that?
I think that's a really good question. I don't think I've got an answer for you. I'm kind of puzzled about what it is. But I think if you look at the other data that's been presented, particularly the maize work, where they did grow, and I don't remember all of the different things that they did, but they grew in different locations. But in some years, the differences were almost minimal. Whereas others, there was a more significant increase. And this isn't actually that unusual. If you think of normal breeding processes where they would take a range of different new varieties and put them out into different locations and different trials, then there is variation from year to year. So I think the jury's out on whether the tobacco plants were not limited in field conditions. We have already shown by overexpressing other enzymes in tobacco, and there's been fairly substantial pieces of work done there, that you can actually improve photosynthesis. But whether the process that ictB is involved in, that's somewhat still in a black box, was not limiting in that year under those particular circumstances, then we have to, it's a bit of arm waving really, but that does look as if it's probably down to the environmental conditions. I mean, I think tobacco is also quite sensitive, actually. Its roots are very sensitive during the early growth season. So if something had happened then, that might mean it would have lost any advantage that it had. So I don't have an answer for you, but it's a really good question. And I think it's important that we talk about these things. I think it's really important to realize that not everything is successful. And when in the greenhouse, and when you transfer it to the field, there's no guarantee that it's going to work.
Okay, so Chaim's got a follow-up question. Given that the initial slope of the ACI curve was not affected even in past work, does this give any clues to the function of ictB?
Yeah, well, I mean, of course, it was very exciting when the very original paper did show that VC max was changed in those early transformations, and I have no idea, why they would have observed that and nobody else has. There is really very little. Maybe I think maybe was it Will Hayes? There might have been a small hint in soybean that there was some increase in that initial slope, but I think we certainly haven't seen it in our tobacco plants, and I did not observe it in the Arabidopsis work that was done either. So you are right, yes. Had we seen that, that would still hint to the fact that although it's maybe not directly impacting CO2 uptake, it may have an indirect effect. But I think right now we really don't know what it's doing. It looks like it's on the microsomal membranes. I mean, and we did do this. We looked at this many years ago when Andy Simpkin put a YFP labeled protein into tobacco here, but we thought that we just hadn't done it properly. But I think quite a few other people now have also shown that it's not in the chloroplast, and it looks as if it's definitely in the cytosolic membranes.
Okay, thanks, Christine. So Hazel has a couple of questions. I think the first one follows up a little bit. Hazel asks, you mentioned a previous paper showing different impact of ictB transformations in different locations. Could you touch more on that and how these results relate to your own study?
Well, I think the way that it relates to me is I think it highlights the fact that we only did the study 1 year in the field in Illinois, and that might not be where that would have had the most benefit given the environmental conditions at that time. I think the work that was done was obviously in maize. That's a C4 plant. So the impact ictB was having there might well be different. It looked, if I remember correctly, to be expressed in the bundle sheath cells, but not in the chloroplast. So, that would lend I guess its positive effects to being more with the C3 cycle than with the C4 part of the pathway. But it doesn't give us any more indication as to what the actual reason for that uplift would have been. I hope I've answered that question. I think really the main parallel with our work was that they did it in different locations. I think if they'd only done it 1 year in one location, they may also, I don't know what the chronological order was in the paper, but they may well have not seen an increase. So I hope that addresses that question.
Okay, and Hazel follows up. With the increased number of leaves, could this lead to an increased rate of photosynthesis in older plants?
I mean, that's a really interesting question because, we definitely think the effect of photosynthesis is compound. So often you do see increases in leaf area in the younger plants, and therefore one would envisage that if you manage to get a bigger leaf area and you've got, even if you've got the same rate of photosynthesis across that whole leaf area, then you would imagine that you would get an uplift. What we don't really know about is when we got the increased number of leaves. What I'm not clear about, because it was in the field, it was a destructive harvest at the end of the season, and we didn't follow that through the season to see if, I mean, I don't know if you've seen tobacco when it grows. This is Samson, and what happens as you come towards flowering, you get a whole bunch of rather small leaves up at the top of the plant, and I'm not quite sure if the increased number of leaves necessarily would have led to an increase in a rate of photosynthesis. I think there wouldn't be a direct effect. It just might be you've got more leaves, therefore you get more photosynthesis.
Okay.
Thank you, Hazel, for your questions. Really good.
Thanks, Hazel, for the question. Yeah. Don't see any other questions at the moment, so maybe I can ask a couple. So going back more generally to the RIPE project, where you've been stacking multiple genes that have individually been shown to enhance photosynthesis. What are you generally observing? Are you observing an additive effect, or does the incremental effect of stacking diminish as you put more genes in?
I don't think, John, I mean, it's a good question. I don't think we've done this systematically. I mean, if you look at two of the successes that have been obtained with Steve Long's work on the NP, and Steve Long and Chris Niogi's work on the NPQ, and then Don Ort with his team on the photorespiratory bypass. They really required that number of proteins to be introduced in order to get the effect, and particularly for the photorespiratory bypass in order to complete that part of the cycle. For us, I mean, you're right to ask us that question. I think what we did observe, if you look at our Nature Plants paper, when we put the SPPAs together with the cytochrome C6, it was only when we put the two together we really saw a positive effect. The underlying reasons for that, well, boosting electron transport together with boosting the RUPB regeneration may well give you an additive effect, but not in all conditions. So I think the jury's out. I think we did see, Andy saw very clear effects in the greenhouse, Andy Simkin, with his work in the original JXB paper in 2015, where he put FBPAs together with SPPAs based on a modelling paper from earlier, absolutely got synergistic effects. And there was a really important point there, actually, that I could maybe just, if we've got time, I could just open up a little bit. Because we introduced SPPAs and FPPA aldolase, both involved in the RUPP regeneration part of the cycle. And when we measured photosynthesis in the young leaves, the SPPAs, individual overexpressors, had an increase in photosynthesis then. But when we looked in the older leaves, then SPPAs didn't seem to have the same positive effect. However, when we had SPPAs and FPP aldolase together, we could see an increase in photosynthesis in both the young and old leaves. So you would imagine that that would give at least an additive effect there, and also an increase in biomass. And of course, when we added ictB in, we also saw this additive effect. And I don't know if I could say it was synergistic. I think it's probably additive at the moment. It's not huge, but it's definitely there. I think the other thing about multi-gene manipulations that may offer a benefit is that you may not always see a kind of 1 + 1 + 1 giving you a kind of continual improvement. But what you may get is some resilience that's built in, and so that perhaps deals better with these environmental variables that you come across in the field. And therefore, you would still see an uplift. It may not be as large as you would have predicted from greenhouse experiments, but it would give you that option of resilience.
Okay, thank you. Do we have any more questions from the audience?
Happy to take any kind of questions. Oh, we have got some.
Tamara. Tamara asks, can you detect ictB in cyanobacteria, or only when you express it in the plants, I guess?
You know, I don't have the answer to that question. I could. I mean, I think obviously they're able to detect it because they isolated it from there, but I don't know. I know what you mean. In a Western blot, can they just run that? I don't know. I'd need to look that up. I don't know if there's anybody in the room that knows this work that could answer that. I think I've asked that question. Andy, I don't want to put you on the spot, and I don't know if you're actually there or not, but I didn't look back at that older paper on the isolation of it. I would imagine that would have been done genetically and through molecular biology rather than because you would have had to have raised the antibodies. So my feeling is that although there has been the occasional report of antibodies that can detect ictB, I've yet to have a set of them put into my hands that would enable us to do that. And even expression of the whole protein was really difficult. So I think they definitely can identify the gene, and the absence of that gene has a phenotype in cyanobacteria, but I'm not clear that I don't know that they necessarily would have been able to do that. I think the main reason was we did write to people who had published something with a Western blot in, and I think we got some antibodies and they didn't work. This is years ago, and then more recently again asked. Oh, I think it was maybe it was the Israel group sent me some antibodies, and we couldn't get anything really significant from it. They had trouble as well. They didn't profess to have been able to do that. And so really, I think the only data that is recent is the Mays paper that was published at the end of last year that I showed earlier in my talk. I think that was Kustner et al. And it was a +1 paper. I think that what they did was chop the protein up and managed to do some work with that. So I hope that answers your question a bit. I think I'm fairly sure that I have not seen a cyanobacterial extract on a gel with a Western blot, but I may have missed it.
Yeah, thanks. Hymes put a comment in that in the original paper it was only northern blots. So
yeah, no, I think they were pretty straightforward. They were really helpful, but we were never be able to use it to identify the protein. And I think they also had the similar problem.
Yeah.
Must be tucked up really in that membrane, you know, somewhere. And it could well be that some of the epitopes that you might be able to identify are just hidden.
Yeah.
Yeah, that's another good point. Yep, that's another good, yep.
Levels might be really low and strongly regulated.
We tried a proteomic approach as well. Hussein Gurley tried a proteomic approach to send away extracts and see if we could identify the protein. But of course they couldn't. And of course you need a positive control, and that's when we got into trying to express it. And we weren't able to do that either. I guess what you could do is you could mimic the tryptic digest, I suppose, by creating peptides and using them as a control. But I thought that could be a rabbit hole for someone, so decided that we might not bother going down that one.
Okay, so we're coming to the scheduled end of the session. So there don't seem to be any other burning questions, so we'll close now. So I hope please join me in thanking Christine again